Hi, I am Christine Jacobs-Wagner. I am the director of the Microbial Sciences Institute at Yale University, and I am also a professor of molecular, cellular, and developmental biology and investigator of the Howard Hughes Medical Institute. In the first presentation, I told you a little bit about the cell biology of bacteria and how important it is for cellular function and cellular behavior. In this presentation, I'd like to tell you a little bit about research ongoing in my lab. More specifically, I would like to tell you about a particular project that relates to DNA segregation. So how bacteria are able to segregate the chromosome and achieve active transport. Now, broadly speaking, what my lab is really interested in is understanding how bacterial cells self-replicate. So as you probably know, and it's illustrated in this movie, bacterial cells, if they are put in the right growth condition, they can proliferate at a remarkable pace, something that often we would like to control. And so my lab is really interested in understanding the molecular and physical mechanisms by which bacteria multiply at this fantastic pace. And so to do this, we address a lot of questions such as how do bacterial cells grow, how do they divide, how do they couple these two processes to ensure that they're going to achieve an optimal shape and size for function, how do they replicate the DNA, how do they segregate the DNA, how do they coordinate cell cycle events with cell division and growth. We also want to understand how they regulate gene expression to maximize self-replication. But today, because of time constraint, I would like to focus on one particular question, which is how do bacterial cells partition the cellular content to ensure that each daughter cell is going to get its fair share of cellular component so that it can continue this self-replication process. Now, actually, bacteria, because as I said in my first presentation, they are very small relative to eukaryotic cells. So this is how a bacteria, so I drew here how most bacteria would look like relative to an animal cells. So they're very tiny. And this is actually an advantage because it means that diffusion, which is a passive process, is going to be an effective mean for molecular transport inside a cell. So diffusion, which is driven by thermal fluctuation, is going to work well in bacterial cells because they are small, and diffusion works well over short distances. So this is illustrated in this simulation that we made for you. So just consider a cellular component that is in 100 copies, and all 100 copies are localized inside us in the middle of the cell. So just imagine a protein in 100 copies. Now, as I start the simulation, in very little amount of time, simply due to Bromian motion, so passive diffusion, you're going to have a quick distribution of those 100 copies of the cellular component, and random distribution here, or dispersion of this component, is just going to be good enough for partitioning, for equipartitioning between your daughter cells. So if cell division here happens in the middle, each daughter cell is going to get its fair share of about half of the copies. And so diffusion alone is going to be an effective means for equipartitioning of many cellular components in bacterial cells. So that works well if the cellular component is in many copies. But if you have very few copies of that cellular components, and there is many components in the cytoplasm that are in few copies, now random distribution is not going to work as well. And so here we have the same simulation, but now instead of having 100 copies, we have only 2 copies. And as you can imagine, depending when division is going to happen, there is actually a chance that one of the daughter cells is going to get both copies, and one daughter cells will get 0 copies. And you can do a very simple calculation, which is what we did here, to show that random distribution is going to, for components that are in copy number of 10 or less, there's going to be a significant probability, and this probability is going to increase as the copy number decreases, a probability that one of the daughter cells is not going to get any copy of that component. And so that means that bacterial cells had to evolve a system to ensure the partitioning of cellular components that are in low count. And a very important and versatile system that bacteria have evolved is the ParB system, which we call also the ParABS. And so ParABS systems are important for chromosome segregation. They've been shown also to be important for plasmid partitioning. So some low copy plasmid encode their own ParABS system that is important for the partitioning, such that each daughter cell is going to get an equal number of these plasmid. But ParABS systems have been also important for the partitioning, for the segregation of other cellular components, including enzymatic microcompartment and cytoplasmic chemotaxis cluster. What I'm going to talk about today is the ParABS system that is important for chromosome segregation, and the model system that we use is Caulobacter crescentus. So what is nice about the ParABS system is that it's only made of 3 components. One is the DNA sequence, and the other 2 are proteins. So the DNA sequence is called ParABS. And so ParABS has been shown to be very conserved, to be found on many bacterial genomes, and is typically located close to the origin of replication. ParB is a protein. It's a DNA binding protein that's going to bind to the ParABS sequence and form what we call the partition complex. And then ParA, this is a wild-caught type A ATPase that is going to be important for the translocation of the partition complex. So since we are looking at this in Caulobacter crescentus, I just want to remind you that Caulobacter is basically a typical bacterium in the sense that it has one circular chromosome, it has an original replication here, and the ParABS sequence in Caulobacter, just as in many bacteria, is actually located close to the origin of replication. As I mentioned in my first presentation, the chromosome in bacterial cells have to be compact about 1,000 times to fit into the bacterial cells. And so this is more the way we think it would look like inside the Caulobacter crescentus cells. So the DNA, which I hope you can see, is those blue strands that are packed into the bacterial cells. But as I mentioned in my first presentation, the chromosome is folded in a spatially organized fashion, such that each gene is going to be located at a specific location inside the cells, and such that in Caulobacter, the origin replication is always found at one end of the cells, which we called the old pole, to distinguish it from the new pole at the opposite end, which is called new pole because it was made by the last division. Since the ParABS region is close to the origin of replication, it means that it's also going to be found at that location, at the old cell pole. And as I will show you in a minute, this ParABS region is the first region that's going to be translocated after duplication, translocated at the opposite end of the cells in the process of chromosome segregation. So now to explain what happened during chromosome segregation, I just want to show you some more rudimentary schematics. So now in the schematic, for the sake of simplicity, I drew the chromosome just as this gray line. And again, the ParABS sequence is located at the old pole. And since ParB is going to bind to this ParABS sequence, going to form the partition complex that is shown here in green. It's actually tethered to the end of the cells there because ParB is going to physically interact with another protein called ParBZ, which is located at that end of the cells. So ParBZ is a protein that multimerizes, form a matrix at the end of the cells, and it's sticky. It's going to bind to ParB and thereby tether the chromosome at that end of the cell. So this is the rest of the Caulobacter cell cycle. And Caulobacter has a very interesting cell cycle by having, polar appendages like stalks. It's going to have also flagellum here, and it's going to divide asymmetrically. This is all very interesting features that we study. However, today those are not important because today I'm just going to talk about chromosome segregation, which happens in all bacteria, and it's not specific to a cell cycle. All right, so let's go back here. So at this stage now, DNA replication occurs. And since the ParABS sequence is close to the origin of replication, it means that it's going to be one of the first regions that's going to be duplicated. And therefore, this is going to create 2 partition complexes. One is going to stay at one end of the cells. The other, which is the first step of chromosome segregation, the other is going to be actively transported to the other side of the cells. And this is going to happen in a ParA dependent fashion. And this step, so this active motion from one end of the cells to the other is essential for viability. Okay, so this is actually happening while replication is still ongoing. So in bacteria, unlike in eukaryotic cells, replication and segregation occurs in the same time. So as the partition complex is one of them is migrating to the opposite end, when it reached the end, it's going to tether. It's going to be tethered because it's going to now interact with a growing ParBZ matrix. And now DNA replication is going to keep on going, and the chromosome is going to unzip such that each daughter cells is going to inherit a chromosome following division. Okay, so the partition complex, what is it made of? So this is shown here. So as I mentioned, the ParS region is close to the original replication, which is not shown in the schematic. But in Caulobacter, there's two ParS sequences, which is shown in red. ParB is going to bind to those ParS sequences. And as it binds specifically and recognizes those sequences, now the protein, ParB protein, is going to be able to spread. Some people say that it polymerizes on the DNA. Simply because of those two ParS sequences, when it binds to it, this is going to result in the spreading of the protein on adjacent sequences, regardless of what the sequence composition is. And we can estimate the number of ParB protein that are associated with this DNA sequence. And so by doing quantitative Western blotting, we know the average number of ParB protein inside the cells. And by doing quantitative resonance microscopy, we could establish that there was about 80% of the ParB protein inside the cells that is associated with the DNA, the ParS sequence. And so that is roughly 580 molecules of ParB that is associated to the DNA, that is part of the partition complex. This is important because it means that you're going to have a very strong, very high local concentration of ParB at the partition complex. This is going to become important later on. Comparatively, we have only about 140 molecules of diffusing ParB. So that's just 20% of the pool. And that's going to result in a very low concentration of ParB in the cytoplasm. We estimate it to be under 1 micromolar, whereas like over 500 micromolar at the partition complex. Okay, so we can track the partition complex as it's duplicated and translocated. So because there is so many ParB protein binding to the DNA region, it appears as a bright focus by fluorescence microscopy if you label ParB protein with a fluorescent protein. So when I'm going to start the movie, we're going to have DNA replication occurring that generate a second partition complex. One of them stay at the pole, the other one that is free is going to migrate to the opposite end of the cells. And the movie is going to loop a few times so that you get to see it more than once. So we can quantify this, this motion. This is actually shown here. So we're looking at the partition complex along the cells over time. You see that after duplication, one is going to stay at one end of the cells, the other one is going to be translocated in a ParB-dependent fashion, and then is going to get tethered to the opposite end of the cells. What we can do also is that we can visualize both ParB and ParA in the same cell. And we can do this by tagging them to fluorescent proteins that have a different color. So this is what we did in this experiment. So now what are you basically seeing is ParA is in red, and you also see in green the trace of the partition complex, which is rich in ParB. So this is actually a chemograph where we are looking at the ParA signals along the cell length over time. And just to help you here, we have the schematic. So in the beginning, which is shown here, the ParA signal is basically across the cells, except where the partition complex is. As I'm going to show you in the next slide, it's actually forming a gradient. But then when the partition complex is duplicated, something amazing happens. That is that when the partition complex gets close to the edge of the ParA signal, shown in red, now the ParA signal is going to shrink, and this is going to be correlated with the motion of the partition complex. And this correlation, this correlated dynamics between ParA and ParB, which was first shown in Vibrio cholera for chromosome segregation in that organism, has also been shown to be true for plasmid and other ParABS system. Okay, so let's see this in a different way. So again, here you're looking inside the cells. This is an epifluorescence image. In red, it's ParA. In green is a partition complex. As you can see, there are two of them. One has just duplicated. And now, if you look at it, even if you look at it by super resolution microscopy, you find that the ParA signals across the width of the cells is fairly uniform. But across the length of the cells, it's actually forming a gradient, which is shown here. So here you see along the cell position, you see ParB in green, and you see ParA in red. You can see that it forms this shallow gradient. So this is looking at the beginning, following DNA replication. But if we follow this over time, you can see here that the gradient, the ParA gradient in red, is getting steeper and steeper as the partition complex shown in green is basically moving along as if it was walking along, following the ParA gradient. So how does this happen? What is important for us to know the biochemistry of the system. So the question is, what are the biochemical properties? And here, I'm going to summarize a lot of work by many labs over the years, including ours, with this biochemical model, which was supported by in vitro study and also mutagenesis studies. So what is shown here is ParA. ParA, in the presence, in vitro, if you purify ParA and you put it in the presence of ATP, it's going to form dimers. And then when it becomes a dimer, now it's going to have an affinity for the DNA, any unspecific sequence. So now the ParA dimer is going to bind to the DNA. And it is in this dimeric form that now it's going to have a strong affinity for ParB. So now it's going to bind to ParB, but now after it binds to ParB, ParB is going to stimulate its ATPase activity. So in the absence of ParB, there is very little ATPase activity. I'll show you in the next slide. But what is important is that when it gets in contact with ParB, now this is going to stimulate ATPase hydrolysis, releasing the complex between ParB and the ParA dimer, because now the ParA dimer is going to dissolve into monomer, and then through nucleotide exchange, you can repeat the cycle. Now, what is important in this slide is that, yes, it's going to bind to ParB, and ParB is going to stimulate ATP hydrolysis. But ParB can do this only if there is a high concentration of ParB. And so this is shown in this biochemical experiment. So this is in vitro. So you have purified ParB, you have purified ParA, you add DNA and ATP to your reaction. Now, if you just have no ParB at all, then what we're looking here is the ATPase activity. So we're looking at the turnover of ATP, ATP hydrolysis. Without ParB, you have very little amount of ParA ATPase activity. You do have a little bit, but it's not relevant. It's not high enough to be relevant for the reaction, for the translocation process. But if you increase the concentration of ParB, you see a really nice stimulation of ParA ATPase activity. But this, you get only a significant, relevant ParA ATPase activity at very high concentration of ParB. So you need over 100 micromolar of ParB to get a nice stimulation of ParA ATPase activity. And this is why it's only the partition complex, which is really rich in ParB, where you have hundreds of molecules of ParB, that is able to stimulate ParA ATPase activity, whereas the diffusing ParB, whose concentration we estimate to be under 1 micromolar, well, this is too low to have any effect. So it means that, again, it's only the partition complex, the ParB-rich partition complex, that's going to bind to the ParA ATP dimer, and that's going to stimulate ParA ATPase activity. All right, so this is how we think it looks like inside the cells. So you have the DNA in blue, and you have the two partition complex. One is going to stay at one pole, scattered there by the ParB matrix, and one is going to migrate somehow along this ParA gradient. ParA, which I'm showing you in red here, is basically to represent ParA ATP dimer, because there is concentration of ATP inside the cell is high enough so that ParA is going to bind ATP, bind to the DNA, and it's going to do so away from the partition complex, forming this gradient. We know by quantitative Western blot Western blotting, and we know also by single molecule counting that there are about 90 dimer of ParA. So this is what we put in this drawing. So then we wonder, is it possible that the way that the partition complex in green here is migrating to the opposite end of the cells in a ParA-dependent fashion is because it's simply diffusing, interacting with some of those ParA-ATP dimer along the way, and this perhaps is resulting in kind of a guided diffusion. So to wonder if that is true, then the first thing that we wonder is whether the partition complex, when we move to the opposite side of the cells, does it do it in a linear fashion, or does it do it in a kind of zigzaggy fashion because it has to interact with many ParATP dimers through diffusion? And what's consistent with diffusion, and we have other data that support that as well that I won't have time to show, is that indeed if we track at very high temporal and spatial resolution the partition complex, you see here that this is one end of the cells, this is the other, that it kind of followed this kind of zigzaggy fashion, and that there is really two phases, that there is a slow motion and that is followed by a fast motion. So then we wanted to test, well, is it possible that what happens is that the partition complex is diffusing and interacting with ParATP dimer, and this is sufficient to reproduce the translocation kinetics that we observe inside a cell. So we wanted to test that through simulation. And so we built a model that was absolutely constrained by experimentally derived parameters. So we measure the parameter inside the cells, and then we constrain our model by the value that we obtained from experiments. So we know the diffusion coefficient for the partition complex. We know how many ParATP dimers there are, which are shown here in red. We know how many ParB protein there are. We know that they can interact with multiple ParATP dimers. We know how the ParA gradient evolved during the translocating process. So we put all this in our model. And then what we do is Brownian dynamic simulation. And then we track the translocation of the partition complex over time. So this is an example of one simulation. Partition complex is diffusing, it's exhibiting Brownian motion, interacting with ParATP dimer until ATP hydrolysis occurs. We know the rate from our biochemical reaction that we have applied to the model. Now you see here that in the experiments, within 10-15 minutes, translocation should be complete. But in this case, in our simulation, it's not complete. And it's not just complete, it doesn't work just for that simulation. We've run 1,000 of simulation and then look at the average behavior, which is shown here. The simulation is in blue. The experiment, which is the average trajectory for the experiment, is in black. And you see in the black curve that within 10 minutes, the partition complex has moved to the other side of the cell on average. But this is not the case for our simulation. So it meant that something was missing from our model. What was missing is DNA dynamics. Now, in our model, the ParATP dimer was static. But the ParATP dimer is bound to the DNA, which we know is dynamic. And this is shown here. Here, what we have is one chromosomal region that is fluorescently labeled so we can track it over time. And as you can see here, it's actually moving, it's wiggling around. And this has been known for quite some time. And you can take many different chromosomal regions, different regions on the chromosome, and you see the same behavior. You can quantify this motion, so you can obtain trajectories, and you can analyze those trajectories, which is what we've done here. And we found that the chromosomal loci, so the chromosome, display elastic dynamics. And this is shown here because we see this harmonic potential that imply elastic dynamic. So what it means is that chromosomal loci are basically winging around, but always coming back to an equilibrium point as if they were on an elastic. So now, because ParATP dimer is bound to the DNA, it means that those ParATP dimer are going to experience the elastic dynamics of the underlying DNA. So now we're going to add this dynamics that we are measuring in experiments from the motion of chromosomal loci. Now we're going to incorporate it into our model. So this is exactly the same model, same parameters, but the only difference is that now the ParATP dimer, which is shown in red here, are going to wiggle around, displaying the DNA dynamics that we observe in the experiment. And so we have the same thing. We have this is an example of a simulation. In the beginning, we see a slow translocation. But as it goes through the more dense region of ParA, you see an acceleration of the translocation. Now, the ParATP dimer are not disappearing in our experiments. We are just simply being redistributed as they interact with ParA. They are being redistributed, forming the evolution of the ParA gradient that we see in the experiment. So this is in one experiment and one simulation. We did thousands of simulation, 1,000 simulation. We can average it. And now we see that our model agrees very well with the experiment. So in green is the result from the model, where the only difference is that we incorporate the DNA dynamics. As you can see, that it follows very nicely the experiment showing in black. Okay, so we call this model the DNA relay model, because what we think is going on is that the partition complex, which diffuses when it's going to interact with a ParATP dimer that is moving, that is exhibiting this elastic dynamic, when it interacts with it, it's going to experience this elastic dynamics, bringing the partition complex to the equilibrium point until hydrolysis occurs, being released, and now it's going to be passed on to another DNA region. So basically, the partition complex is going to move from one DNA region to another using ParATP dimer as transient tether. And so this is what we try to illustrate here in this animation. So in this animation, before I start the movie, I'll just tell you what you're going to see. So here you have the partition complex, which is rich in ParB. And then you have ParATP dimers that are bound to the DNA. Now the DNA start to form those plectinine loops, which is shown here. And those loops are basically going to exhibit elastic dynamics. And then as I start the movie, the partition complex is basically diffusing. Then it's going to encounter a ParATP dimer. That's basically, because of the elastic dynamics, going to bring it to the equilibrium point until ATP hydrolysis occurs, and now it's going to be released. And now it's going to interact with one or more ParATP dimers. That's going to bring it to the next DNA region. And the reason we think that it works, as shown in our simulation, is because the partition complex comes from one side relative to the ParATP dimers. That is going to be the probability of being in a stretch configuration to bring it to the equilibrium point. And so together with ex vivo, we created an animation that illustrates what we think happens from the top.
Caulobacter crescentus has a circular chromosome that is condensed into a spatially organized 3D structure inside the cell. The origin of replication and two nearby ParS sequences are located at one end of the cell, called the old cell pole. The DNA-binding protein ParB recognizes ParS and spreads onto flanking DNA. This makes the so-called partition complex. Replication creates a second partition complex that diffuses away. Inside the cell, the DNA is highly dynamic. An elastic force returns each DNA locus to its equilibrium position. ParA, another protein critical for chromosome segregation, binds ATP, forms dimers that associate with the dynamic DNA away from the partition complex. When the diffusing partition complex interacts with one or more DNA-bound ParA dimers, it experiences the elastic force from the underlying DNA. ParB stimulates ParA ATP hydrolysis, leading to the release of ParA monomers from the partition complex and the DNA. Repeated rounds of association and dissociation between ParA and ParB effectively transport the partition complex towards the new cell pole.
I hope you have enjoyed the animation. So in conclusion, the data suggests that the cells is using a DNA relay mechanism in which the cells are harnessing the elastic dynamics of the DNA to achieve directed motion. So in other words, the chromosome is playing a mechanical function. So of course now we have a million questions to address, too many to list, but some of the first question that we're interested in addressing is how is the pi-ATP dimer gradient achieved, and is it really important for achieving this directed motion? Another important question is how general is this DNA relay mechanism? We think that it might be general, that it might be important for chromosome segregation, but it also might be important for other processes that exploit a ParABS system. The reason why we think this is because the elastic dynamics of the DNA that we observe in Caulobacter crescentus are also true in other bacterial cells, such as in E. coli. And it has also been shown that they are true in eukaryotic cells, where chromatin display elastic dynamics in nuclei. So it is likely that those elastic dynamics of the chromosome is a universal properties of DNA that is in a confined space. Furthermore, every time it was tested, it was shown that the DNA binding activity of ParA is conserved, which would be important for this DNA relay mechanism. And so to end, I would just like to acknowledge the work that was done by, the experimental work was done by Hong-Shun Lim and Whitman Schofield, whereas the Bromian dynamic simulation was done by Ivan Suretsev and Bruno Beltran in my lab. In addition, I would like to thank all the members, past and current, of my lab for their really nice contribution over the years. And in particular, I would like to thank Brad Perry, who was a graduate student that made the two first simulations that I showed in the beginning of the talk. And of course, I would like to give a big thanks to the National Institute of Health and the Howard Hughes Medical Institute for their generous supports over the years. And I would like also to thank you for your attention.